And we're live, and welcome to another episode of the Machine Medicine interview series. We are delighted and fortunate to have Chandramouli Krishnan, who is a assistant professor at the University of Michigan in Ann Arbor, in a number of different areas. And actually, Chandramouli, that would be great to start off with just understanding a little bit about your background for our listeners.
Definitely. Thank you very much, Jonathan, for having me. I'm basically physical therapist by background. So I got my bachelor's in physiotherapy in India from Chennai. And then I practiced physical therapy for almost 6.5 years, primarily initially in outpatient sports medicine setup, and then went into spinal rehabilitation, and then actually directing PT department in a super specialty hospital. And then I moved to University of Iowa, where I got my master's and PhD in sports medicine and biomechanics. Again, primarily focusing in the area of knee injuries and knee rehabilitation. And then did a postdoc. That's when I took a turn away from my sports medicine and biomechanics background into neurorehabilitation, robotics, and non-invasive brain stimulation for rehabilitation. That was at Rehab Institute of Chicago, which is now called as Shirley Ryan's Ability Lab. And then in 2012, I joined University of Michigan as an assistant professor, and where I also founded the Neuromuscular and Rehabilitation Robotics Lab. The short acronym for it is NeuroLab with a double R. And I'm currently an associate professor at the University of Michigan Physical Medicine and Rehabilitation Department.
Fascinating. So a couple of things in there. One thing is interesting to observe that you changed fields essentially. Can you describe a little bit what was it that sort of either disillusioned you about your original field or enticed you into this one? What made you think this is the kind of exciting area you wanted to dedicate your career to?
Yeah, and again, right, like I do both now. So I do both sports medicine, biomechanics, and also neurorehabilitation and neuroplasticity. So the reason I moved from my sports medicine area to a different branch is just to get, postdoc is an opportunity where one can get a diverse skill set and then try to merge the two areas. So one thing I wanted to do, and which I've been doing successfully, is merging the field of orthopedic and neuroscience into a somewhat field called as orthopedic neuroscience. So basically taking elements from neuroscience and applying it in the orthopedics, and same way taking elements from orthopedics and applying in neuroscience. So that's why I went out of my current area what I was doing in my PhD, and then broaden my skill sets so that I can, be not so tunnel viewed and keep doing what I was doing in PhD, but rather expand my area of research work.
Fascinating. A theme that we often come across, that the most exciting research and opportunities are often at the interface between two sort of intrinsically interesting areas, but that seems to be the interface is often the most sort of exciting place to be. So really interesting. It would be good to understand a little bit also, Chandramouli, about the tools of your trade. What sort of, these are such large areas, what sort of niche, as it were, within this area did you decide to sort of focus on? What are the tools of your trade generally?
Yeah, so my research work primarily spans across the field of engineering, rehabilitation, motor control, biomechanics, and neuroplasticity. And so my lab does, and it's fascinating. Our work involves a lot of benchtop device development to cross-sectional, more basic science mechanistic questions, to large-scale clinical trials where we use these tools that we have developed and then test it on long-term interventions to see the efficacy and effectiveness of the tools that we have developed. And along those lines, and we have developed like a lot of, the primary central theme of my lab is low-cost technologies for rehab. So in general, as you may know, it's US is primarily third-party payers, so insurance cost is very critical for covering rehabilitation services, and oftentimes it's very hard to penetrate into the rehabilitation clinic if the tools that are available for rehabilitation are very expensive. And so that's what, like, even for my postdoc, like, I was doing a lot of, $500,000 kind of robotic tools. And then I moved away because I quickly realized, like, it is very hard. Big centers like University of Michigan can afford, but, like, most of the regular clinics cannot afford those kind of technologies, and patients, most of them don't come. A good portion come to University of Michigan, but you can see majority of them are treated at smaller outpatient clinics or subacute rehabilitation facilities, which cannot afford these large expensive robotic devices. Plus, these tools cannot penetrate into patients' home if you want to extend the dosage of therapy. So that's kind of the motivation for me to move along the area of how can we make these technologies low cost, and then make it portable, lightweight, and can be carried home. So that has surrounded my work. And so in our lab, and along those lines, we also do a lot of work related to both neuromodulation, but also evaluating neuroplasticity and brain physiological changes, after a intervention with a robotic device, or when how people interact with robotic device, what kind of neuroplastic changes that it can create at the brain level. So from neuromodulation side, I worked in the past a lot in transcranial direct current stimulation, which is a very low cost device. And then we also use TMS both as an evaluation tool, but also as a neuromodulation tool, where we actually use a principle called as operant conditioning of motor evoked potentials to prime the brain and also to improve corticospinal plasticity.
Okay, let's talk about that for a bit, if that's okay. What is transcranial magnetic stimulation? How does it work? What are the principles underlying it? What do we think we're doing when we use it?
Yeah, so transcranial magnetic stimulation is basically a magnetic stimulator that sends out magnetic field into your brain, and then it gets converted into electrical energy, and then it sends action potential to stimulate certain regions wherever you are stimulating. And if you think about your brain, all of your connections to the muscles are all in the brain, right? And so lower extremity has some regions in the brain, and then you have a motor map of all of the muscles in the primary motor cortex of the brain. And so when you stimulate these regions, you can measure the responses that are coming from the brain to the muscle by putting an electromyographical sensors on the muscles of interest. The other form of looking at it is, which is what we also do, we published some recent papers on it, is we evaluate the torque responses that are associated with those stimulation. And so basically, you can use this TMS as kind of two broad tool. One is an evaluation, which is basically to see the excitability level of the brain, and that is using a single or paired pulse, which is two pulse. You give some separation between the first pulse and second pulse, and there are a lot of protocols that are there to evaluate the inhibitory and excitatory circuits in the brain. The other form of TMS is basically repetitive TMS, where you keep giving repetitively the transcranial magnetic stimulation pulses at certain frequency, and there is a wide variety of protocols that are there in the literature. And you can either upregulate the brain or downregulate the brain. And if you think about stroke, oftentimes, and one can argue against it, but there is a good literature that suggests that the lesion hemisphere has down regulation of the corticospinal excitability, and there is some upregulation in the non-lesion hemisphere in terms of corticospinal excitability. And so people have tried various forms where they can either upregulate the lesion hemisphere or downregulate the non-lesion hemisphere. with various protocols and look at it.
Interesting.
Yeah.
So maybe I can just unpack a little bit of that. So just to go back to the sort of basics, or other people that haven't used TMS, I never have, for example. But the basic sort of principles underlying it are that the brain uses, nervous systems in general use electromagnetism as their language of interaction, as it were. And one can talk to populations of neurons, as it were, at least interact with them by switching an electromagnetic field on and off very rapidly using electric current, I take it. So it's an electromagnet, and use that to interact with certain circuits at the level, presumably fairly close to the sort of skull, the cortical region, the sort of as it were, kind of orange skin of very complex neuronal material that seems to encapsulate the brain, and then you say also there's some obviously this makes sort of sense because your brain controls your muscles, and this is the whole feeds into the whole neurorehabilitation kind of field. But then also you said that there's some evidence now that in major problems such as stroke, we all know what a gargantuan issue that is as a health challenge, and also therapeutic challenges. But there's some evidence that this may be a good way to interact with nervous systems that have been the subject of an insult like a stroke. What kind of, so let's just go back to that. Hopefully we've got a sort of picture of how that works. What kind of resolution are we talking about interacting with neuronal populations here? Is it's presumably quite a few now, and at least initially was, but in terms of the potential of the technology, how fine-grained a sort of needle could we achieve with this technology ultimately in the sort of the general prospects?
Yeah, it is a very great question. And if you think about any of the non-invasive brain stimulation technologies, they lack spatial resolution in general, right? Like you cannot be very specific. Just because if you really think about it, the whole motor cortical strip itself is very small, right, and narrow, and it's not like it's not the entire brain, right? Like different regions control different parts. And so when it comes to focality, transcranial magnetic stimulation is more focused, at least spatial resolution wise, than transcranial direct current stimulation, conventional transcranial direct current stimulation. But even in TMS, the spatial resolution, like say for example, if you are focusing on lower extremity, which is what we do a lot, is you need to go deep into the brain, right? It's in the central sulcus, most of the lower extremity representations are. And so when we stimulate it, we are going to get stimulations in multiple muscles. It's not going to be very specific to the muscle that we are targeting. And one way we circumvent that problem a little bit is by giving a background contraction. So what happens is, say, if you want to stimulate the tibialis anterior, which is a muscle that controls the foot to lift it up, if you wanted to focus on that muscle, one way to focus is if you make them to slightly contract their muscles, then what happens is it's like you can think it as a path of least resistance kind of. So your motor neuron pool is already excited. That's connecting the tibialis anterior from the brain. And so what happens is now when you stimulate it, most of the impulses go to the muscles of interest. So that's one way of improving the focality. But there is also another issue, and if you are aware of something called as neuronavigation, so we can use it's a way of capturing where that coil is in the brain, right? And so the issue is, in general, when we use transcranial magnetic stimulation, which is a big coil that are placed, once we find the location, even if you slightly change the orientation of the coil, right, or the position, it doesn't have to move a lot. Even if it moved by half a centimeter, right, it's not a lot, but completely you will be in a different region, and your evaluations could be affected by where you are. And so one way people circumvent that problem is by using an approach called as navigated transcranial magnetic stimulation, where people put markers. There are various kinds that are available, but the most common form is they put a cluster of marker in the head and then cluster marker in the TMS coil, and then map the, head system to the coil system. And then people can also use MRIs from the brain and then somewhat map these different regions and then say, okay, you are in this region, and it gives you feedback on your software, which tells like which part of your brain you are at, and then you can use that as a feedback to be at least somewhat precise, at least within the day, right? Even between the day, but there's slightly more error between the day, because every time you come in, you have to co-register all of these points, right? And so there's going to be some error in co-registrations. And so within day, it's a lot more reliable, and even it improves reliability between days.
I see. So the anatomical precision is hugely important.
Yep.
How about the complexity of the kind of temporal patterns of stimulation? Is it typically, you mentioned a few things like the short interval pulses, stimulation, that's one thing that you do, but in other areas such as DBS, there's interesting avenues of research in doing things like stimulating stochastically and this kind of stuff. Is there a parallel avenue of research in TMS, or does it tend to be very sort of periodic and stereotyped patterns of stimulation?
Currently, most of the paradigms that are used are very stereotyped, and again, when I say stereotyped, perhaps like each group may have their own stereotyped patterns of stimulation, and they stick to it kind of. But in general, right, like most of the TMS protocols are very stereotyped in terms of the, temporal timing and things like that is very fixed. And the reason is it could be like, prior work would have eluded them to use certain parameters. And if you start tweaking the parameters a lot, it becomes challenging in terms of replications of studies and things like that. So it is stereotyped as of now. There are labs that are trying various other parameters, and especially that happens in transcranial alternating current stimulation, which is another form of portable stimulation technique, where they tend to, it's an alternating current, so they tend to focus on, alpha rhythms, beta rhythms, and different rhythms that the brain have, and they try to tweak the parameters more related to those things.
Yeah, I see. So I'll ask one more question if that's okay about this, because I just think it's so interesting. Could this potentially be a paradigm for a kind of, the project that Neuralink is engaged in is, trying to establish a sort of information bus between the outside and the inside of the brain, and they're doing it by having an actual physical neuronal sort of machinery interface. Other companies and other researchers are trying non-invasive approaches, some using optical techniques. Is this another candidate in that sort of field for setting up an information exchange point, a kind of USB of the brain?
So you were saying, can you rephrase your question? So if I understand your question, you're asking, can this be a potential tool going forward in the field of rehabilitation?
Well, there's a desire, if you think about the founding sort of thesis of Neuralink, the idea is that if we could increase the ability to exchange information, as it were, increase the bandwidth of the exchange mechanism between neuronal tissue and what artificial hardware, sort of between mind and machine, as it were, then we could obviously treat lots of conditions, and I think that Neuralink, they'd be interested in developing sort of consumer applications, but let's stick to the sort of medical sort of applications. And the hope is that essentially that, as it were, USB for the brain could be used to reprogram nervous systems that are in a state of unhealth back into a state of health, in principle at least. But then the question is, what exactly does that USB look like? Is it, as Neuralink are supposing or hypothesizing, an actual physical sort of metal on neuron kind of interaction, or is it a non-invasive system that's based upon, for example, the optical characteristics of neurons? I don't need to tell you, doubtless there's a lot of work now exploring those, either with things like voltage-sensitive dyes, or just the optical properties of neurons as they change as they fire action potentials, for example. And I was wondering, is there anything fundamental about the, as it were, magnetic sort of route that would prevent that from potentially being another sort of avenue worth exploring?
Yeah, one of the issue with the transcranial magnetic stimulation is the hardware. There is a hardware limitation. So the coil has to charge and discharge. So if you wanted to, like, if you really think about some of the neuroprosthesis where people put in a chip into the brain and then try to use the neuronal signals that are coming out to encode that signal and then control
Exactly.
a robotic arm, it is very feasible in that kind of neurotechnologies because the temporal resolution is very good. In the sense, when I say temporal resolution, even TMS has temporal resolution, but in terms of the ability to take information and then also do something to control, the problem with TMS is once you trigger a pulse, right, you need to have some gap, and you cannot, you can, like with high frequency TMS, you can stimulate in a much faster, but then the issue over there is it creates somewhat of a unsafe paradigm where it could start reducing the seizure threshold and create seizures and things like that. And so there is an established protocol in terms of um, how fast you can go and beyond which is not safe. So to use TMS as a tool to as a two-way system is kind of little hard, and how people have circumvented that problem is they use EEG along with TMS. So they take information from EEG and then try to stimulate, but still TMS has not been, at least to my knowledge, has not been used in a way where it's a continuous modular waveform kind of where you continuously tweak the parameters based on the signals that are coming out from the neuronal population. And the real reason for that is, apart from safety, there are also hardware limitations in terms of how you can use that.
Interesting. So let me sort of turn that around. What is the greatest sort of therapeutic potential of TMS then, do we think? Would it be the stroke application that you mentioned before, or are there other even broader approaches that might be taken?
Yeah, and again, if you really look at, I mean, it's FDA approved already for depression, and there has been good evidence in terms of people who don't respond to medications and they are refractory. They do tend to respond well for people who have depression. They do tend to do well with transcranial magnetic stimulation. So it has been FDA approved. A lot of clinics use it, but in terms of neurorehabilitation, there is a lot of work that needs to be done because it's not necessarily just for TMS. This goes more actually into transcranial direct current stimulation technologies, and that's one reason I have slowly moving away from TDCS while I've used it in the past is the issue of reliability and replicability of the study results. And often see we all work on the premise. The basic premise is, while there are many scientific premises, the basic premise is these neuromodulatory tools like TDCS or TMS alters brain excitability. And you can control it to either upregulate or downregulate the excitability so that then that transforms into, when paired with some sort of behavioral interventions, it transforms into better outcomes and better functions and better recovery. But the issue is the ability of neuromodulatory techniques to uh, reliably induce changes in corticospinal excitability has been questioned a lot recently, because if you see 5 studies that show tremendous results, there is equally or more studies that show no effect or null effect. So the field of neuromodulation has to do a lot of work, and which is good challenge to the neuromodulation community, because they can unify, and somewhat they have to be device agnostic, right? Like, just because they have used certain protocols in the past, um, it's time for everybody to rethink. Okay, like, let's as a field unite and come up with paradigms that have shown very reliable results. Because if we want to make insurance company to reimburse, and they're going to look at solid evidence, and right now we don't have solid evidence towards one side or the other. And so we need to really work on that before we can make it as a promising tool. But I do see, especially TMS modalities, have definitely some good potential in terms of neurorehabilitation. And what we don't know is timing, right? Like, when should we give this therapy, right? Like, there are a lot of uncertainties that we need to first understand: dosage, timing, how can it be paired with other treatment modalities and things like that. And the nice thing is NIH, which is our kind of primary funding agency, National Institute of Health, has established a center called as National Center for Neuromodulation for Rehabilitation, which is NCNM4R, which is a center that actually trains people who are interested to go into the field of neuromodulation, do a lot of workshop, and most of the researchers over there are somewhat device agnostic. They don't really are married to one device versus the other. They're just trying to be impartial and look at the a field in general, how we can move the field forward, right? And once we have that, I think once we are focused down, and once we know what works and how can we reliably induce and replicate those results, I think we are in a good situation to make it as a more clinical tool to impact recovery after stroke or other neurological disorders, right? I mean, if you really think about spinal cord injury, the neuromodulation has gone fantastic, right? Like, but it's not non-invasive, but currently there are results that are from non-invasive transcutaneous electrical stimulation, which are showing somewhat good benefits even for completely spinal cord injured patients. Now, I'm sure you may be familiar with a lot of work from Grigor Cotain, Rijay Jitton's lab, and others in the US, where they have shown that people who are completely paralyzed after spinal cord injury, if you implant them with implantable electrodes and use neuromodulation and train them, I mean, that's most crucial, is you cannot just simply rely on the neuromodulation. You have to always pair it with some sort of physical therapy or behavioral interventions. Once you do that, and they have shown that people are able to regain some of their lost function, and they can even walk after complete spinal cord injury using those neuromodulatory techniques.
That's fascinating. Would it be fair, if I can try and summarize one of the lessons from that, then would it be fair to say that there's something of a crisis in terms of standardization between labs and sites, and then maybe we could finish by talking a little bit and briefly, I'm sorry that we haven't got as much time as we perhaps like, but about some of your work that you've done in trying to sort of create a software approach to standardizing the deployment of TMS devices. Could you say a little bit about the motivation for that? And the
Yeah,
basic?
definitely. So the one of the issues, and this is not just for neuromodulation. If you take motor control, the same issues there. If you take other fields in general that are rehabilitation or neuroscience oriented, standardization has always been a problem. And in fact, one of my colleagues at MSU had a recent beautiful article about how the field is fragmented in terms of the tests that they use for motor control. Same way, like, we have, like, if you take TMS, we have like so many paradigms, right? And so everybody is kind of, oh, this is my paradigm, kind of, right? And so the field is fragmented, and because of which it's very hard to replicate, right? Like, because if there are 60 paradigms to 100 paradigms, and there is always very hard to replicate everything, right? And so field as a whole, it's very hard for us to replicate. And one way of standardizing things, and I did that, and that was one of the motivation, one way of standardizing things from the software side is, at least, if we make the tools available open source, perhaps people will start using, then it's the same tool that is used. At least you can narrow down to one less variable that we have to worry about, right? And if you see that a lot of our work, we tend to make it as an open source software or hardware that is available to the public. And I guess you are alluding to the work that we have done in neuronavigation, where we have created a couple of tools for navigated TMS, which is initially we attempted that process using a webcam, two webcam, and then using some sort of triangulation method to know the location of the markers, and creating models to pretty much replicate the um, uh, navigated TMS that are available in the market. But we had, I mean, like, it was good, but we had some challenges, and then we moved to a low-cost camera system, which was OptiTrack, which has actually, it's a 3D camera, so we don't have to worry about other calibration procedures and things like that. And then we used a Unity software engine to basically take information that are streamed out from the OptiTrack camera system, and then create a tool in terms of making uh, um, coordinated access system for the coil, and then the model the head. And then we created a very nice software that provides feedback on where your coil is with respect to the head. And this work was actually done, and this is where I feel like, there are a lot of things that can be achieved with at very low cost. Actually, an undergraduate student, Daniel Kotmeyer, who actually worked with me to help build this. Initially, it was done by another undergraduate student, Jacob Rodseth, and then Daniel Kotmeyer finished it. And we were, in fact, trying to incorporate MRIs. The only thing that it misses now is we don't have imaging system incorporated into the software. We can still, like, if we are interested in motor evoked potential, it would still do a very good job of providing feedback, because oftentimes people who use motor cortex for stimulation, they would tend to just rely on motor evoked potential. So you can find the initial location, and then tell, this is the spot I want to be, this is the orientation I want to be, give me feedback on that, and it provides feedback. The only issue with that is we still don't know where your brain is if you wanted to target other brain regions, right? Because if you wanted to focus on dorsolateral prefrontal cortex or some other brain region, cerebellum, we just don't get motor work potential. So we need to rely on the MRI systems to give us that information. And unfortunately, while Daniel was working on this, but he passed away. So we kind of put an halt in the development process. But eventually, I'm going to seek out some funding, and hopefully we will advance on that front.
I'm sorry to hear that, but some really wonderful work. And so there were a couple of themes that you brought out there. One of them was that getting away from expensive hardware, and then another one was the power of software to perform these kind of co-registration tasks. And then another one was, of course, there's a clinician deployment, which is a source of variability. But as you mentioned too, this is at the anatomical location is an incredibly sensitive parameter, and there's a huge amount of anatomical variation between individuals intracranially, which you can't perceive from the outside. But there does seem to be the potential for software to make some big contributions here.
Yep.
Great. Cool. Mauli, I am sorry that we've actually overrun slightly, but it was a pleasure to do so because it's such an interesting field, and you're making some great contributions to it with your students and your colleagues. So my congratulations on that, and thanks again so much. We'll keep a close eye on your work, and I do hope to catch up again soon and see what you're up to in the near future.
Thank you very much, Jonathan. It was nice talking to you. Thank you for this opportunity.